The process of fertilization here, it is called double fertilization and triple fission. It is the transfer of pollen grain from anther of one flower to the stigma of another flower, but of the same plant. Then it contains 3 cells at the chelygel end, which are called as antipodal cells, which will degenerate. So what is the weightage of marks for 3 marker question? For 8 questions, it is 24 marks. Hello everyone. In today's session, discussing about a very important chapter 1 of 2nd PUC, that is sexual reproduction in flowering plants. So I'll be briefing up about this particular chapter, which has a lot of weightage of marks, that is sexual reproduction in flowering plants. Myself, B. Shobharani, from the Department of Biology, Vidhyaashram Pre-University College, the Temple of Excellence in Mysuru. In today's session, before I move on to the revision of this very important chapter, let me brief up about the blueprint of the 2nd PUC theory paper, because we all know that there is little bit changes that have been done in the syllabus. So there are some chapters which are deleted, and what will be the weightage of marks for the chapters that are included in the present syllabus. So let me move on to this very important slide, which will give you an idea about the question paper of 2nd PUC biology. Moving on to this very important slide, as we know, the question paper consists of these many parts. Part A1, Part A2, Part B3, Part C4, Part D. In the previous paper, it was section 1 and section 2. Here it is Part D, 5th and 6th. So what will be the pattern of question type that is being given in the present question paper of 2nd PUC biology? As we all know that the first very important is the multiple choice questions, that is MCQs, where you will have around 15 questions. The number of questions that are given for MCQ is 15. So what will be the weightage of marks? Obviously it is 15 marks. So the next is, that is A1 will include what? A1 is MCQ, 15 questions, 15 marks. A2, fill in the blanks, that is 5 questions, 5 marks. So part A1 completely deals with 1 marker question, that is 1 marks, that is MCQ and fill in the blank. That you must know that part A1 and part 2 includes 1 mark. Moving on to part B, that is the third part, which is having the short answer type of questions, and the weightage of marks is 2 marks. So part B means it has 2 marker questions. So how many questions are asked? That I am talking about this whole question paper is including the choice questions. So you'll have around 8 here. The weightage of marks for 8 is 16. I am talking about including the choice questions. Then moving on to part C, that is the 3 marker questions. That is short answers, that is 3 markers question, which is number of questions that appear in the question paper is 8. So what is the weightage of marks for 3 marker question? For 8 questions, it is 24 marks. So part A, A1, 15, part A2, 5, part B, that is 8, part C4, that is 8. That is 15, 5, 16 and 24 marks. Moving on to part D, 5th main, that is long answers for 5 marker question. You will have around 8 questions in the question paper. The weightage of marks for 8 will be 40 because it is 5 marker question. And part D, that is long answers, 5 marker question, that is 3 will be there. 3 questions appear in the question paper and the weightage of marks is 15. So the total number of questions that is going to appear in the 2nd PUC biology question paper is 47. Previously it was 37, but now as we know it has changed. The pattern has changed. So it is 47. So what will be the weightage of marks for 47 questions, which includes the choice questions, is 115. Here it is 47. Here it is 115. So one, that is MCQ, 15 marks. Then fill in the blanks, 5 marks. Then 2 marker questions, 16 marks. 3 marker question, 24 marks. 5 marker question, that is part D, 5th question, that is 40 marks. And again part D, which is again a long answer question type, that is for 5 marks, which is 15. So total it is 115 marks. So 47 questions will appear in the question paper. This is very, very important point to be noted. Now let us discuss about the chapters that are included for the second PUC biology syllabus that is there for your exam and the weightage of marks. As I said, I am revising the first very important chapter of second PUC biology, that is sexual reproduction in flowering plants. So what is the weightage of marks for this particular chapter? It is 11 marks. So it's having a very huge marks of weightage. So we need to concentrate much on this particular chapter called sexual reproduction in flowering plants. So MCQ, you will have one question. Then again MCQ, like you'll have two questions here. Then short answers 1. Then again MCQs 1. So totally you'll have that is long answers 1 question. So here it is 11 marks. Human reproduction, that is the second chapter, is having a weightage of 11 marks. So again same, short answers 1, then MCQ 1, long answers 1, then MCQ 1 and again MCQ 1. So total 11 marks. Reproductive, the third chapter, has a weightage of 6 marks. So sexual first chapter 11 marks, second chapter that is human reproduction 11 marks, then reproductive health 6 marks. Then here you can see MCQ 1, short answers 1, that is essay 2, that is short answer to 1, then MCQ 1. So total it is 6 marks. Moving on to principles of inheritance and variation, where the weightage of marks for this particular chapter is 14 again. You can make out from this slide itself a very important slide which will give you a brief outlook of the pattern of question paper and the weightage of marks. Here itself you can make out sexual reproduction in flowering plants is having a weightage of 11 marks and principles of inheritance and variation is having a weightage of 14 marks. So these two chapters are having lot of weightage. So you students have to concentrate on these two very important chapters. Again, so what are the type of questions you are going to get here? Short answers 1, then MCQ 1, long answers 1, then again MCQ 1. So total it is 14 marks. So here, then moving on to molecular basis of inheritance, 14 marks. Again, this particular chapter is also having a lot of weightage. So here it is 14 marks. So evolution 7 marks, then human health and disease 13 marks, then microbes in human welfare 8 marks, biotechnology principles and processes it is 8 marks, biotechnology and its applications 6 marks, then organism and population 7 marks, then ecosystem is 5 marks, and biodiversity and conservation it is 5 marks. So total weightage of marks is here it is 115. As I already said, the weightage of marks is for the whole question paper including the choice is 115 and the number of questions that are going to appear from part A to part D is 47. So this is a very, very important slide which you need to understand from the examination point of view. So once again I'll repeat. Sexual reproduction in flowering plants 11, human reproduction 11, reproductive health 6, principles of inheritance and variation 4, molecular basis of inheritance 14, evolution 7, human health and disease 13, microbes in human welfare 8, then biotechnology principles and processes 8, biotechnology and its application 6, organism and population 7, ecosystem 5, biodiversity and conservation it is 5. So total weightage of marks for the whole syllabus is 115, which includes, as such you all know that your paper is for 70 marks, but the whole question paper will include 115 marks, which includes even the choice questions. Let me move on. So I hope I am clear with this particular slide. So let me move on to the revision part of a very important chapter, that is sexual reproduction in flowering plants. So even one more slide I have added here so that you will get to know how the second PUC question paper pattern will be. So already I told you the question paper consists of 4 parts A, B, C and D. Part A1 consists of 15 multiple choice questions. Part 2 consists of 5 fill in the blanks questions, as I already told. All the questions of part A1 and 2 are to be answered compulsorily. So you cannot miss out. As you know, 20 marks are from part A itself, which is compulsory. Then part B consists of 8 short answers carrying 2 marks each, out of which you are going to answer how many questions? 5 questions. Then part C consists of 8 short answer type questions carrying 3 marks each, out of which 5 questions to be answered. Part D, as I already said, consists of 2 sections like part 5 and part 6 of part D. Part D, fifth, consists of 8 long answers type questions carrying 5 marks each, out of which 4 questions to be answered. Then part D, that is 6, consists of 3 long answer type questions carrying 5 marks each, out of which 1 question to be answered. So this is the second PUC question paper pattern. Hope I am clear even with this slide. So moving on to the synopsis of a very important chapter I told, as I said in the beginning, the first chapter of your second PUC biology syllabus, that is sexual reproduction in flowering plants, which is having a weightage of 11 marks. So what are the points to remember in this particular chapter, which is very important from examination point of view? Stamens, as we all know, the male reproductive part of a flower. As we all know that flowers are the marvels and embryological marvels of the plant kingdom. So as I said, the stamens are the male reproductive part of a male reproductive units, you can call. Then microsporangium, organ which produces the stamen or the pollen grains, and microsporogenesis, the process that involves the process of formation of the pollen grains from pollen mother cells through meiosis. Then structure of pollen grain you all will be studying. Development of male gametophyte, that is the pollen grain. Then pollen allergies. So as we all know that pollen grains are also used as a source of food in many cases. At the same time, it is also causing allergies. Then use of pollen grains, as I already said. Then we'll be talking about the very important female part of a flower, that is pistil or gynoecium. As we all know that the pistil has 3 parts: stigma, style and ovary. Then megasporogenesis, the process of formation of megaspore from megaspore mother cells through meiosis is called megasporogenesis, and process of formation of embryo sac or female gametophyte. Characteristic distribution of cells in the embryo sac, as we all know, we call it as 8 nucleate 7 cell stage. Then pollination, which is a very important phenomenon which needs to take place to bring the 2 gametes together. That is pollination, which is nothing but the transfer of pollen grains from the anther of a flower to the stigma of a flower. So there we'll be studying about the types of autogamy. In that again, chasmogamous flowers and cleistogamous flowers, the conditions that are required for autogamy. Then geitonogamy and xenogamy. Then agents of pollination. So we also know that pollination can happen only through agents. The one which will help in the process of pollination may be abiotic agents or biotic agents. So abiotic agents include wind and water. Then pollination by wind, as we all know, it is called anemophily. Pollination by water, it is called hydrophily. Then biotic, that is the agent that is helping in the process of pollination is the living organisms, that is mainly the animals. And thereon a very important event has to take place, that is pollen pistil interaction. So once the pollen grains land on the stigmatic surface, thereon the events that happen till the process of fertilization, you call it as pollen pistil interaction. Then fertilization, that is the fusion of the male gamete with the female gamete, resulting in the process of formation of the zygote, and the fusion you call it as syngamy and triple fusion. Syngamy and triple fusion. That is why the process of fertilization here, it is called double fertilization and triple fusion. I think all this you have already studied in theory. So in this particular session, I'm going to revise. I'll make you understand briefly as to what are all the important concepts you people are going to study in this particular chapter called sexual reproduction in flowering plants, which is having a weightage of 11 marks. So moving on to the multiple choice questions. As already said, Part A1 consists of multiple choice questions. Around 15 questions are asked, which is for 15 marks. As I said, Part A1 and Part 2 is compulsory. Just have given 2 examples to make you understand like what type of questions will appear for MCQs. For example, like the first MCQ, the most resistant organic material present on exine of pollen grain. As we all know, pollen grain has two envelopes or integuments, outer exine and inner entine. So the most resistant organic material present on exine, what is it? Is it A, option A, tapetum, option B, germ pore, or sporopollen and cellulose? So the option is what? Sporopollen. So as we know, it is highly resistant organic material present in the exine of pollen grain. Next, second question. It's just an example I have given, like that. Good many number of questions, MCQs are going to appear from, as I already given you the idea about the blueprint, like how many MCQs questions you all are going to get from this particular chapter. So second MCQs, just for example, tassels in corn cob represent. What are the tassels in corn cob represent? Is it ovary or anther, fifth month, stigma and style? So the answer is what? What does tassels in corn cob represent? Stigma and style. So option D is the right answer, stigma and style. So this is how the MCQs appear from this particular chapter. So I've just given two examples to make you understand what type of MCQs are going to appear from this particular chapter called sexual reproduction in flowering plants. So all the questions what I have given in this particular session of the chapter called sexual reproduction in flowering plants includes the questions that appeared the March 2023 and up to March 2014. So I have included all the questions, the possible questions that were asked from March 2023 to March 2014. So like what type of questions were asked? So like for example, the third question, differentiate between geitonogamy and xenogamy. This is like a most possible question that is asked in the paper. Why? Because this is a very important word called geitonogamy. Because what is geitonogamy? It is the transfer of pollen grain from anther of one flower to the stigma of another flower, but of the same plant. So more or less, it is like cross-pollination, but it is like self-pollination. Because you see, it is the transfer of pollen grains from anther of one flower to the stigma of another flower of the same plant. Then what is xenogamy then? Transfer of pollen grains from anther to the stigma of different plant. Same plant, geitonogamy. Different plant, we call it as xenogamy. So this is a very, very important question which is asked in the examination. So differentiate between geitonogamy and xenogamy. Next, this is a long question. As I said, all these questions what I have framed is from the questions which were the previous question papers from March 2023 to 2014. So what is double fertilization? Describe fertilized embryo sac with a neat label diagram. So I already told you the type of fertilization here, it is called double fertilization and triple fusion. So here, describe fertilized embryo sac with a neat label diagram. So let us see what is this question number 4. What is double fertilization? Describe fertilized embryo sac with a neat label diagram. What is double fertilization? The phenomenon of two types of fusions. What is that fusion called? Syngamy and triple fusion that take place in an embryo sac of flowering plant is termed as double fertilization. The fertilized embryo sac consists of, see here they have given described fertilized embryo sac with a neat label diagram. The fertilized embryo sac consists of large primary endosperm cell consisting of triploid endosperm nucleus. It also consists of diploid zygote. At chalazal end, it consists of degenerating antipodal cells. As we know, there are two ends. That is the chalazal end and the micropylar end. So at the chalazal end, there are three antipodal cells which will degenerate. Degenerating synergids at its micropylar end. So at the micropylar end, there is one called the zygote as well as the degenerating synergids. Next, differentiate between geitonogamy and xenogamy. Already have explained. That is, geitonogamy is the transfer of pollen grains from anther of one flower to the stigma of the same plant. Transfer of pollen grains from anther to the stigma of a different plant. Define polyembryony. What is polyembryony? Poly means what? Many. Embryo. So, occurrence of more than one embryo in a seed is referred to as polyembryony. Question number 7. Mention the organic resistant material present in the exine of pollen grains, that is sporopollen. Question number 8. Define the following terms. Staminate flower. Unisexual male flowers bearing only the stamen. Stamen means it's the male reproductive part of a flower. Pistil, as we know, it is a female reproductive part of a flower. That is unisexual female flowers bearing only the pistil. So what is staminate flower? Unisexual male flowers bearing only the stamens. Pistillate flower, unisexual female flowers bearing only the pistil. Question number 9. Differentiate between albuminous and non-albuminous seeds. The seeds which retain a part of endosperm as it is not completely used up during embryo development is called albuminous seed. Then non-albuminous, what are the examples you are going to give for albuminous seeds? Wheat, maize, barley and castor. Non-albuminous seeds. The seeds which have no residual endosperm as it is completely consumed. So here some amount, here it is used up during some amount, it will retain. Some amount of endosperm is retained. Here it is completely consumed during embryo development is called non-albuminous. Example, pea and groundnut. Moving on to question number 10. Cleistogamous flowers are invariably autogamous. Justify the statement. This is a very, very important question. Usually they ask in the examination. So you can put a star here. It's a very important question from examination point of view. Why cleistogamous flowers are invariably autogamous? It's one of the conditions for autogamy also. In cleistogamous flowers, the anther and stigma lie close to each other. When anthers dehisce or open in the flower buds, pollen grains come in contact with stigma to affect pollination. So as there is no chance of cross pollen landing on the stigma, so obviously cleistogamous flowers are invariably autogamous. That is the reason this is a very important question from examination point of view. So this is a very beautiful diagram. Define megasporogenesis. Describe the internal structure of a mature embryo sac of an angiosperm flower. So this is the diagram. Here you can see there is two end, chalajal end and the micropylar end. So what do you see in the chalajal end? 3 antipodal cells. Here you can see the polar nuclei, 2 polar nuclei. Then this is the central cell and this is the egg and these two are synergids. And a very, very important thing you can see here, that is the filiform apparatus, which will enable the pollen tube carrying the 2 male gametes enter the embryo sac and help one of the male gamete to fuse with the egg cell to form what? Zygote. So this is the structure of an embryo sac. So what is megasporogenesis? The process of formation of megaspore from the megaspore mother cell, MMC, through meiosis is called what? Megasporogenesis. Structure of mature female gametophyte or matured embryo sac. This is a very important diagram from examination point of view. So here you can see each embryo sac has 2 poles, as already said, chalajal pole and micropylar pole. Then it contains 3 cells at the chalajal end, which are called as antipodal cells, which will degenerate. Then 3 cells at the micropylar end. Here you can see the 3 cells. Among the 3 cells, 2 are synergids and 1 is the egg cell. And together, what do you call this whole thing as? This whole thing is called as egg apparatus. Egg apparatus, which includes how many cells? 3 cells, 1 and 2 synergids, including the filiform apparatus. So here, the synergids have special cellular thickenings at the micropylar tip called filiform apparatus, which play an important role in guiding the pollen tubes into the synergids, as I already said. Then in the center, very important, there is a large central cell with 2 polar nuclei. So already the 2 polar nuclei is 2n, and one of the male gamete will come and fuse with the 2 polar nuclei to form endosperm nucleus. So this is 3n. Hence, because the process of fertilization happens 2 times, here and here, it is called double fertilization and triple fusion. That is what I said. Why it is called as double fertilization and triple fusion. Thus, a typical angiosperm sac at maturity has what? 8 nuclei and 7 cell stage. Then, moving on to question number 12. Name the type of pollination that brings genetically different types of pollen to stigma. As we all know that flowers prefer cross-pollination than self-pollination. So in order to avoid inbreeding depression, they'll come out with certain strategies also. So what is xenogamy? The type of pollination that brings genetically different types of pollen to stigma. Then 13. So as I already said, this is one like a most common question which is asked. Sporopollenin is one of the most resistant organic material known. Justify. Why sporopollen is one of the most resistant organic material? Because the sporopollenin, one of the most resistant organic material known to cause it, that is, any strong acid and alkali will not have any effect on the sporopollen. So no enzyme that degrades sporopollenin is far known. Because of this property, pollen grain can be preserved as what? Fossils. That is the reason why there is no as such organic acid or alkali which can degenerate or degrade sporopollenin, and that is the reason why the pollen grain can be stored for a longer time. Then 14th question, differentiate between staminate and pistillate flower. Which part, and it's a split question for 2-mark question, which part of the flower produces fruit after fertilization? So what is staminate flower? Unisexual male flower bearing only the stamens is called as staminate flower. Pistillate flower means what? Unisexual female flower bearing only the pistil. Then moving on to the next question, question number 15. Describe the structure of dicot embryo with the help of a label diagram. So this is the structure of a dicot embryo. So which is also very important diagram from examination point of view. So maybe for 3-mark question only the diagram might be asked, but for 5-mark question along with describe. So here they have again describe the structure of the dicot embryo with the help of a neat label diagram. So what is dicot embryo consisting of? It consists of an embryonal axis with 2 cotyledons, hence the name dicot. And the portion of the embryonal axis above the level of cotyledon is the epicotyl, which terminates with the plumule or stem tip. So this part. Then the cylindrical portion below the level of cotyledon is known as hypocotyl. That terminates at its lower end is the radicle or root tip. Here you can see the radicle or the root tip. Here it is the plumule. Then the root tip is covered by root cap, which is like a protection. Then 16th question. Explain any 3 outbreeding devices in flowering plants. That is unisexual flowers. So usually a plant tries to bear unisexual flowers to avoid what? Self-pollination and to enhance cross-pollination. So what do you mean by outbreeding devices? The devices are the strategies that a plant will come out to avoid self-pollination and to enhance cross-pollination. So here unisexual, so if both male and female flowers are present on the same plant, so usually self-pollination is possible. It prevents autogamy but not geitonogamy. Male and female flowers are present in different plant dioecious which prevents autogamy and geitonogamy. Example, papaya. Dichogamy. In some species, pollen release and stigma receptivity are not synchronized. So there are 2 different times. So to prevent self-pollination and favors cross-pollination. Proto-anther is one of the outbreeding device. Flower in which anther mature earlier to stigma to prevent what? Self-pollination. Example, sunflower and cotton. Then protogyne, flower in which stigma matures earlier to anther to prevent self-pollination. Self-sterility or self-incompatibility, very, very important. Only when the stigma is receptive, then only it is going to accept the pollen grain that is going to fall on the stigmatic surface. As we all know that stigma is the landing surface for the pollen grains. Thereon the pollen and pistil interaction is going to happen. So the pollen grains do not germinate on stigma of same flower due to mutual inhibition. In this, the genetic mechanism prevents the pollen germination on stigma. Moving on to question number 17. How many chromosomes are there in myocytes of human beings? That is 46 or 23 pairs. Name the part of the flower which develops into fruit after fertilization, that is ovary. Question number 19. Define autogamy. Write the 2 different kinds of flowers that exhibit autogamy. So it is the transfer of pollen grain from anther to stigma of same flower of the same plant. Example, oxalis, viola and commelina. 20th. Explain a mature embryo sac with a neat label diagram. So here you can refer March 2022 question paper wherein they have asked this question. Explain a mature embryo sac with a neat label diagram. As I already said, these are the most probable questions that were asked from March 2023 to March 2014. What is apomixis? Formation of seed from an ovule without fertilization. Question number 20. This 22. This is the differentiation between pericarp and perisperm. This is also a very, very important question which is asked for 2-marker question. Pericarp. Fruit wall is called pericarp. What is perisperm? Persistent nucellus is called perisperm. Example, you can give black pepper. So this is a very important question. In most of the question papers it is asked. So it is very important question from examination point of view. Moving on to question number 23, which is a very important diagram. Very, very important. It is like the most probable question that is asked for the long answer types or even for the, but it is asked with a neat label diagram, describe the TS of young anther. So it is for 5-marker question. If only diagram was asked, it was for 3-marker question. So this is the diagram of the transverse section of an young anther, that is the microsporangium, where you can see the 4 megaspores, where you can see producing the pollen grains. You can see the epidermis, endothecium, sporogenous tissue and the tapetum and the middle layers, what I said here. And if this is enlarged, you can see epidermis, endothecium, middle layers, microspore mother cells. As I said, what is microsporogenesis? The process of formation of microspores from microspore mother cell. So it is then the tapetum. Now let us see the description part. Structure of microsporangium. In a TS, as you know, it is appears circular in outline. It is surrounded by four wall layers called what? Epidermis, endothecium, middle layers and tapetum. So outer 3 layers, what is the function of the 3 layers? Epidermis, endothecium and middle layer. Only protection and help in dehiscence of anther to release pollen grains. The innermost layer, very important, tapetum. What is the function of tapetum? It nourishes the developing pollen grains. Cells of tapetum possess dense cytoplasm and have more than one nucleus. In young anther, microsporangium contains sporogenous tissue at the center which undergoes meiosis to produce microspore tetrads. So you can see the 4 there, which further develop into pollen grains. Question number 24. What is embryogenesis? Mention 2 important events that occur during embryo. This is a 2-marker question. So it's like a split question. What is embryogenesis? 1 mark. Mention 2 important events that occur during embryogenesis. The process of development of an embryo from the zygote is called embryogenesis. 2 important events that occur during embryogenesis is cell division and cell differentiation. Question number 25. Write 3 advantages offered by the seeds of angiosperm. So what is the advantage of seeds? Dispersal to new habitats and help the species to colonize in other areas is one of the advantage. Sufficient. So it is for 3-marker questions. 3 advantages. Each advantage carries 1 mark. Sufficient food reserve nourishes young seedlings. The hard seed coat provides protection to young embryo. They generate new genetic recombination leading to variations. Then question number 26. What is megasporogenesis? In the previous slide, what is microsporogenesis and the structure of the TS of young anther? Explain the development of 8-nucleate embryo sac in flowering plants. So what is megasporogenesis? The process of formation of megaspores from the megaspore mother cell is called megasporogenesis. In a majority of flowering plants, one of the megaspores is functional while the other 3 generate. Only the functional megaspore develops into female gametophyte, that is the embryo sac. This method of embryo sac formation from a single megaspore is called monosporic development. Very important term. Then the nucleus of the functional megaspore divides mitotically to form 2 nuclei which move to the opposite poles, forming the 2-nucleate embryo sac. 2 more sequential mitotic division. I think in the previous slide you can see the diagram here. Then formation of the 4 nucleate and later 8 nucleate stages of the embryo sac. After the 8 nucleate stage, cell walls are laid down leading to the organization of the typical female gametophyte or embryo sac. In the observation, you can see 6 of the 8 nuclei are surrounded by cell walls. So hence we use the word called organized into cells. The remaining 2 nuclei called polar nuclei are situated at the central region. See here you can see the diagram. 2, 4 and 8 nucleate stages of embryo sac and a mature embryo sac. Here you can see the diagrammatic representation of a mature embryo sac where there are 2 poles. One is called the chalazal end or the chalazal pole, micropolar pole or micropolar end. And 3 cells that are towards the chalazal end, you call them as antipodal cells, which degenerate, and the central cell has the 2 polar nuclei. And towards the micropylar, there are 3 cells, where 2 are synergids, and here 1 is egg cell. And this is the filiform apparatus which will facilitate the pollen tube carrying the male gamete to take its entry into the embryo sac. This whole unit you call it as embryo sac. So this is the structure, diagrammatic representation of mature embryo. This diagram is very important from examination point of view. 27th question. Write the function of filiform apparatus. I said this is very important term. Filiform apparatus play an important role in guiding the pollen tubes to penetrate into the synergids. Write any 4 characteristics of the flowers pollinated by wind. So pollination by wind is called anemophily. So wind pollination requires that the pollen grains are light in weight. This is also a very important question from examination. So this is usually asked. What is the nature of the flower that is pollinated through wind? So wind pollinated flowers, pollen grains should be what? Light in weight. They should be non-sticky because they'll just fly in air, and powdery form so that they can get distributed everywhere, so that they can be transported easily by wind current. Then they often possess well-exposed stamens so that the pollen grains are easily dispersed into wind currents. The flower must have feathery stigma, very important, so that the feathery stigma will receive the pollen, easily trap the airborne pollen grains. Wind pollinated flowers often have a single ovule in each ovary and numerous flowers packed into inflorescence. Moving on to question number 28. So here you can see the structure of the pollen grain, that is outer exine and inner entine. Already I told you, exine is made up of a chemical substance called sporopollenin. And you can see that actually the exine is a discontinuous layer, intine is a continuous layer. So here you can see there is a germ pore, where actually it helps in the dehiscence. Then the generative cell and the vegetative cell you can see. So structure of pollen grain. Pollen grains are generally spherical and measuring about 25 to 50 micrometer in diameter. It has 2 prominent layers, outer exine and inner intine. Exine is made up of sporopollenin, as you know, which is one of the most resistant organic material. It can withstand strong acid and alkali. No enzyme that degrades sporopollen is so far known. Because of this property, pollen grains can be preserved as what? Fossils. The intine is thin and continuous layer made up of cellulose and pectin. A mature pollen grain consists of 2 cells, namely vegetative cell and generative cell. The vegetative cell is large with abundant food reserve and large irregularly shaped nucleus. The generative cell is small and floats in the cytoplasm of vegetative cell. It is spindle shaped with dense cytoplasm and nucleus. Usually the pollen grain is shed at 2 cell stage in 60% of the angiosperms. In the remaining species, the generative cell divides mitotically and a 3-cell pollen grain is shed. Moving on to question number 29. Draw a neat label diagram of a typical Anatropous ovule. This diagram is very important from examination point of view. So Anatropous ovule, they'll ask. So this is the diagrammatic view of a typical Anatropous ovule, which is having a hilum here, funicle, micropylar end, micropylar pole, outer integument, inner integument, nucellus and embryo sac. So you need to learn this diagram with labeling. Labelings are very important. If you draw a beautiful diagram but you don't label the parts, unlabeled diagram, unlabeled illustrations carry 0 marks. So make sure once you draw the diagram, write the labelings on one side of the diagram. So here, explain the structure of megasporangium. That is the same Anatropous ovule they have explained here. So the ovule is small structure attached to the placenta by means of a stalk called funicle. Here you can see funicle. The body of the ovule fuses with the funicle in the region called hilum. Here you can see the hilum. And the ovule has 1 or 2 protective envelopes called integuments. Here you can see the outer integument and inner integument. These encircle the ovule except at the tip you can see where a small opening called micropylar is there. So here you can see the micropylar end. Then the chalazal end is present opposite to the micropylar end, representing the basal part of the ovule. So this is the basal part of the ovule. The integuments enclose a mass of cells with reserved food called nucellus. So here you can see the nucellus. That is this part. So here you can see inter-integument, outer micropylar end. The nucellus encloses the embryo sac or female. gametophyte. So moving on to 31st question. Papaya plants exhibit xenogamy only. Why? Because they are dioecious. Male and female flowers develop in different plants, hence the name dioecious. Question number 32. How does pollination achieved in Vallisnerian seagrass? So as we know, pollination, there are agents which bring about the process of pollination, and there are two such agents. One is abiotic agents, which includes wind and water. So pollination through wind is called anemophily and pollination through water is called hydrophily. So this is a question which is related to how does the process of pollination happen in Vallisneria and seagrass. So pollination in Vallisneria. Pollination in Vallisneria. So what is so typical about this plant which grows in water, a hydrophyte? In Vallisneria, the female flowers reach the surface of the water by the long stalk and the male flowers released onto the surface of the water. They are carried by water currents and some of them even contact with female flowers and its stigma to bring about pollination. Then how about seagrass, which is also called Zostera? So it is a little bit opposite. Here you can see in seagrass, female flowers remain submerged, whereas there you can see the male flowers floating on the surface of the water, and the pollen grains are released inside the water. They are on the surface of the water. Here it is inside the water. Pollen grains are long, ribbon-like, and they are carried passively slowly through the water current. Some of them reach the stigma and achieve pollination. This is a very, very important question from examination point of view. We can put a star for this. So here in this particular paper session, I have discussed the most likely questions that might be asked in the examination, the coming examination of 2024. So that is the reason why I tried to explain in this particular session the important concepts that are important from examination point of view. And this is one such question which is very important from examination point of view. Moving on to the last but one question. Describe the outbreeding devices that prevent autogamy. Already I have explained, because these are the most likely question which is asked. As I said, that is the reason why again I have repeated. Unisexual flowers, male and female flowers are present in different plants. That is one outbreeding device. Dichogamy in some species, pollen release and stigma receptivity are not synchronized. So to prevent what? Self-pollination and favors cross-pollination. Protoandry flower in which anther mature earlier to stigma to prevent self-pollination. Example, sunflower and cotton. Protogyny flower. See, that is called androecium matures prior to gynoecium, or gynoecium matures prior to androecium. These are the two very important strategies or the outbreeding device which the plant tries to come out with to avoid self-pollination and cross-pollination, to enhance or to favor cross-pollination. Hercogamy, very important, again one of the outbreeding device, flowers in which there is physical barrier between anther and stigma, so which hinders self-pollination so that it can avoid self-pollination. Heterostyle, so in some other species the anther and stigma are placed at different positions so that the pollen cannot come in contact with the stigma of the same flower. So this is one outbreeding device. Self-sterility or self-incompatibility, as I have already explained in the previous slide. The pollen grains do not germinate for mutual inhibition, and in this the genetic mechanism prevents the pollen germination on stigma. And this is the question, the last but one question again, that 34, draw neat label diagram of a LS of a typical flower. So this is the diagram of a typical flower, which has, here you can see the anther, the male reproductive part of a flower. As already said, flowers are the marvels, they are the morphological and embryological marvels of the plant kingdom of the plant. So here you can see the male, and this is the female reproductive part of our flower. And style, as we all know, style, stigma, ovary is the part, stamens, these are the stamens. So as we all know, the four whorls of a flower are what? Calyx, corolla, androecium and gynoecium. So calyx and corolla, the units of calyx are called as what? Sepals. The units of corolla are called as what? Petals. The units of androecium are called as what? Stamens. The units of gynoecium are called as what? Carpels. So as we all know, calyx and corolla are called non-essential orals of a flower, or they are called as accessory orals, which will not take part in the process of reproduction, sexual reproduction directly. So here androecium and gynoecium are called essential orals, or they are otherwise called as reproductive orals, which will directly take part in the process of reproduction. So all these four orals or parts of a flower make a flower complete flower. Okay. Hope you have understood the important concepts of this particular chapter, chapter 1 of your second PUC biology syllabus, called sexual reproduction in flowering plants. So I have tried my best to make you understand the type of questions that are going to be asked in the examination, and also in the first slide I have made it very clear about the blueprint and the question paper pattern and the weightage of marks for each chapter that is being distributed in your second PUC biology syllabus. Thank you.